welcome to another edition of Reptile Fight Club. I'm Justin Julander. Here with me is Mr. Rob Stone. How you doing?
Hello. Great.
Right on. No ahoy hoy this time, huh? All right. Well, today we got another great guest. Nick Vine is here, so we're excited to have him on and chat with him a bit. Thanks for coming on, Nick.
Yeah, absolutely.
All right. So I guess, we've been in contact over the years, a bit, kind of here and there. And you're doing some really cool stuff. So why don't you tell a little bit about yourself, kind of where you're from and how you got into reptiles, maybe briefly, and then what you're doing now.
Yeah. It's nothing new in terms of how I got into reptiles. It was like I grew up watching Steve Irwin. I will say, so this ties into me later on, but I grew up going to Australia because I have family over there.
Cool.
So, my parents lived there before I was born for a couple of years.
Yeah.
And so we have some friends in Sydney, and, some of my earliest memories were handling a big Burmese python at the Sydney Reptile Park. And, there were water dragons in our friend's pool and stuff. But yeah, and then along with that came, I was never really like in the reptile scene. I was very much in my own little bubble. I didn't know how to really go herping. I just kind of incidentally found whatever I could find. And when I was 11, my mom finally gave in and let me get a bearded dragon. And then it was lots of negotiating and it was always no snakes, but lots of negotiations later. I've had like frilled dragon and chameleons, and right now I've got a fairly small collection, but been all over the place. Red crested geckos and bearded dragons, and now I'm in Australia for university. So that's where I've been for the past year and a half, and I'm home right now. But
so is there like a break in the semester or whatever system?
Yeah. So this is my summer break.
Oh, okay.
Because it's reversed.
Summer there. Yeah.
So
you get to come home to cool weather and
Yeah.
Where are you from then? So, or where are you living now, I guess?
Yeah. I'm in just north of San Francisco.
Okay.
So yeah. And then in Australia, I'm at Macquarie University in Sydney.
Okay, cool. That's awesome. Is there a pretty good herpetology scene there at Macquarie?
It's honestly, it's not known for it by any means, but, Rick Shine is there. So I found that out recently.
Oh, really?
I actually didn't know that when I applied. And then now actually, next semester, I've been talking to his lab assistant about potentially helping him, on a project, which is like kind of neat.
That's cool. Yeah.
But yeah. But I didn't know that going into it. I'll say that, but we have a behavioral biology lab that uses Igurnia and nice. some other cool stuff.
Mm-hmm. Awesome. Well, that's really cool. So what are you studying then? Is that your project on Igurnia and sociality, or?
So right now I'm actually just an undergrad.
Okay.
So I'm doing, So my degree is biodiversity and conservation. Mm-hmm. Where that goes is yet to be determined. I may or may not choose to do a master's degree. One of my professors is kind of pressuring me to like get better grades and try to go for that, but we'll see.
Nice. Yeah, we tend to do that.
Yeah.
Hey, have you thought about research?
Yeah. That's cool. I know. He's like, well, you can do what you love for the rest of your life, and maybe you'll even get a museum job. And like, there's possibilities.
Yeah, that's for sure. I always wondered what would have happened if I would have gone down that path, because I was originally slated to be a herpetologist and get my degree there. And then I met my wife, and she's like, how many jobs are out there for herpetology, and what kind of pay are you going to get? And then I started thinking, like, do I really want to pickle stuff? Do I want to, I saw there was
definitely a consideration.
There was this grad student that was moving. I think he'd finished a PhD, and he was, got a job somewhere. And so he was taken off, and he had these cool pets, that, and my old business partner, Ben Morrill, was taking care of him for him. And so I got to see his collection kind of thing. And then I found out when he moved, he just pickled everything. Just
Jesus.
like, yeah, that's like, oh my gosh, that's hardcore. So
I can't really imagine. I mean, yeah,
yeah.
Jesus.
So not the best, but so
Yeah,
now you're
and
kind of, oh,
yeah.
go ahead. Yeah.
Oh no, I'm just thinking about because there is the behavioral biology lab.
Mm-hmm.
That's not really what they do.
Yeah.
So whether or not I'd actually do like the traditional herpetology or more, sociality and reptiles is
Yeah.
something that really interests me. But I don't know. I need to think about it a lot more.
Yeah. Well, it seems like those kind of specifics are going away, and it's more like
Yeah.
we use this model as this, to study this certain system or
Right.
certain, behavior or whatever, or genetics or,
Yeah.
like family groups, you can study
Right.
hernia or you can study bird or, whatever.
Yeah.
So,
Yeah.
so yeah, I guess that's, part of the new, new thing. So, but yeah, that's really cool. That's fun to be over in Australia. I'm so excited. I'm headed over
cool.
in a month and a half here. So
yeah, I know. I've heard.
Yeah. Yeah. I thought maybe Lucas would join us, but he's not, he's local to you, right? He's over there.
Yeah. He's actually, so the Pilbara rock monitors that I did, they're now with him.
Okay.
So yeah,
that's where you got them. Yeah.
Yeah.
Yeah. They're so cool. That's one of my favorites for sure.
Oh, they're awesome. They're heartbreaking, but they're really cool.
Rob prefers the Kimberly rocks, and I don't know. I'm kind of split. I thought I really liked those Pilbara rocks so much better, but I guess once you see them both in the wild, you're like, it's really hard to choose. Yeah.
Well, hey, the Kimberleys that are up in Kakadu, those are definitely up there. But otherwise, I think Pilbaras are where it's at for me. I just can't. The red and especially when you get a really nice contrasty one, they're beautiful.
Now, I was thinking like the Kimberley habitat would be totally different than the Pilbara rock habitat, but like I was up in always the name
Kununurra.
Thank you, Rob. Rob's my second brain, so he remembers words that are not coming to my mouth. So, but yeah, Kununurra is kind of like sandstony, similar to. Well, I guess Pilbara is not really sandstony much either. So
yeah,
more like the.
I haven't been out there yet. Yeah. So, but when I envision the Pilbara, I think I envision just like Kirigini and the big red rock gorges and whatever. Yeah. So,
yeah. There's some great areas out there. Let me know when you're headed out, and I'll see if I can.
I'm trying to line something up.
Cool. Yeah. So you've done a few herp trips while you're down there?
One major one, and then I've got some coming up.
Cool. So you go with like people from the university or herpers or.
The one trip that I did to South Australia was with, I don't know if you might know him, Frizy or Nesty.
Oh yeah.
He's he was great. I just messaged him and we started talking, and he was super nice. And like I mentioned, wanting to go out and find stuff, and I had been there a year and a half and hadn't really gotten out to do any major herping yet, so I was like, it's about time. But yeah, otherwise I'm starting to meet a couple of people locally, but it's been kind of a gradual, like getting to know the area, getting to know people. I've been going to AHS meetings. So it's, I think this semester is where it's really going to kind of come to fruition. I've got a couple things.
That's cool. Yeah, that's very cool. Well, it doesn't hurt to be where Shine is. Read his papers and you'll get a lot of, good stuff there.
Yeah.
I had a chance to meet him and I chickened out. I didn't approach him.
I still haven't met him. I've only met his lab assistant.
Yeah.
I'm in lab meetings with him, and I'm like a little bit, I've talked to him about mimicry in a lab meeting once, because that was like, because the other thing I'm into is Chianactis, and so it was like but anyways, I haven't spoken to him yet.
Nice. Yeah, I love Chianactis as well. Seems we share some interests here.
Yeah.
They're cool snakes. We found a couple out in Southern California there on a trip Rob and I were on, and I found some on a couple other trips, but yeah, they're cool.
They're
snakes.
Super neat.
Yeah. Yep. Well, so you said you had a bit of a collection. Do your family watch it, or you farm it out to other folks, or?
Yeah, kind of both. So yeah, Lucas has my my monitors,
Mm-hmm.
trying to breed them at the moment. I got one clutch, and then my male died, unfortunately.
Gosh, yeah.
So that's a bit of a bummer, but I had a spare, so we'll see.
Mm-hmm.
And then I've got, and then yeah, my mom has been a huge help. She's not super psyched on everything, but she, I've got a tegu, and she likes the tegu, and
Uh-huh.
I also my only snake aside from the Kyanactus I have an anaconda, and she's not so psyched on that.
Yeah.
But yeah,
Nice. Oh, that's cool.
and some other odds and ends.
Sounds like you got a supportive family. I was lucky to have the same thing when I was growing up. I had rattlesnakes and all sorts of stuff that my parents let me keep. So they were really, really
they're definitely not that cool, but but yeah.
Yeah. Well, they probably shouldn't have let me have them. I guess I just too whiny or something. They're like, shut that kid up. Let him have what he wants. No, that's cool. Well, I don't know. I've seen, I think one of my womas may have ovulated recently, so I'm excited about that. Hopefully I switched up their caging and it seems to be having the desired effect. I guess we'll see when eggs come. So that's the key, if the eggs hatch out and are all looking good. So yeah, hopefully we'll have a good season here. And so yeah, I think the blackhead female is also ovulated and getting ready to lay maybe in a month or two. So we'll see how that goes again this year. And I don't know, sometimes those Aspidites, they just break my heart, but we'll see if I can right the ship. I feel bad for all the successful Aspidites breeders listening to me complain about it. They're like, it's not that hard, dude. But we'll see what happens. Yeah. Rob, how's everything going at your place?
Yeah, all good. I mean, we're in the winter wonderland, although we've now had 2 days this week where the storm transitioned from winter wonderland to class 3 kill storm, it seemed, and unexpectedly. So that's been exciting. But yeah, it's everything's, low key here for sure.
Man, it's mild here. We need a little bit of that snow coming our way, but it's just cold. So I always say if it's cold, it might as well snow. But yeah, just dreaming of Australia. That's all I can do in the winter.
Right. Well, at least you have it coming up though. So that's good.
Yeah. That'll be fun. I've been looking at some new areas in Alice Springs. So I'm excited to explore some new stuff and get back. So it's like, since 2010, that was the first time I went to Alice Springs. And so it's been a little while, and I'm, doing my research and looking at all the spots and saying, oh, I have been there. Or I've seen that. Oh, that's where that was, cause it's kind of faintly in your memory. You have a picture of it, but you forgot where you took the picture, and you're like, oh, that was such and such gorge or whatever. So it's nice to kind of put things together. I can't for the life of me find where I put my notes from that trip. I took these notes from the 2010 and 2011 and I think even the 2013 trips, and can't find them. It's driving me nuts because it's got like, places where I found pygmy pythons in Western Australia or, that kind of stuff.
Right.
I want those, the records or the GPS coordinates for those finds, just so I know where I found them, But anyway, so yeah, it's been fun prepping for that and seeing a lot of good targets out there. Right? What's that?
The prep is half the fun. So.
Yeah. And looking at it, I'm like, how did I miss seeing a Stimson's python? Like they're seemingly everywhere out there. There's like one stretch of road where the records are just a solid line along the road. It's crazy. So hopefully I'll get to see a central Stimson eye. That would be nice. All right. Well, we are here. Nick brought up a topic, and so we said, you want to come on and fight it? And he was nice enough to come on the following week. So that was, we appreciate you being on here, and we're going to talk about subspecies and whether or not that's a reasonable classification status. So we will flip the coin between Rob and myself to see who gets the honor of fighting you, and then we'll go from there. So, okay, Rob, you know the drill.
Tails.
It is tails. You win the flip this week. So,
oh, interesting.
Would you like to fight or put your opinion on both sides?
Yeah. So I'm torn because last week, so it hasn't come out yet, right? When we talked to Jordan Parrott,
Yeah.
and you guys had such a good thing going that I didn't want to jump. I'd taken the sort of moderator or both sides role, but I think I'll do it again because on this topic even more clearly, I feel kind of for both sides on this. So yeah, I'll be game either way.
Okay, sounds good. All right, Nick, go ahead and call it for your side there.
All right, heads.
I'm the double loser this week, so nicely done. You want to take pro subspecies or con subspecies?
I think a lot of my research has been in the con subspecies category, so I'd like to argue that side.
Okay, I'd like to hear some good arguments for that because I've got mixed feelings as well. Of course, I'm sure we all do. So it'll be good to kind of flesh out the topic for sure. So as most listeners have figured out by now, this is not truly knockdown, drag out fight. It's more of a discussion and a gentlemanly agreements between friends. So So as the coin toss winner, do you want to defer and let me go first, or do you want to kick it off?
I'll defer.
Okay. All right. So I guess maybe the case for me lies with just classification in general, right? And I guess it probably also depends on which taxonomy scheme or which taxonomist you're following, because it can vary from, Varanus that has all these crazy disparate lines of varanids versus, some of these other ones where they're, dissecting out species from, that probably shouldn't be species in another classification scheme or whatever. So I guess my main thing is I think because I've, done so much with carpet pythons, that's kind of what's drawn me to that subspecies debate, whether or not there should be subspecies. And I could see it going either way, but I think the thing that drives me nuts is when they say it's all just carpet pythons, like from the tip of the Cape York down to diamond python, they're just all the same. And I'm like, I don't believe that. So in that regard, where they're trying to say everything's Morelia spilota, then I say there needs to be a more finer breakdown of what that means and where, those kind of things are. And, looking into that and looking at, some genetic work that's been done, as well as various biogeographical barriers and things like that, kind of led me to the conclusion is there are differences, especially between something like an inland and a diamond and a, southern coastal carpet. So that led to, our kind of go at the taxonomic scheme of carpet pythons. And I think if I were to go more drastic, I might even say that diamond pythons are a different species from southern coastals, from inlands, that I could be just happy with that as well. So I guess maybe I don't necessarily think there has to be subspecies, but in certain cases, I think there probably should be something more fine than just they're all carpet pythons, that attitude. So that's kind of where I'm coming from. Yeah.
It's funny. So I don't know carpet pythons as well as I know, like it's funny that you bring up Varanus, because Varanus is kind of what sent me down this rabbit hole.
Yeah.
Because, so one of the things this isn't, I'll say this and then I'll go more about my view on subspecies, but Varanus are kind of known for having subgenera, which I am not a fan of, because the way that I gather taxonomy is it's all kind of centered around the species. Like the species is the basic unit of taxonomy. And we can argue all day long what is a species and what isn't a species and how do we classify species and is it genes or morphology or whatever. Mm-hmm. But at the end of the day, like a genus is, as I understand it, the next grouping essentially above a species in terms of like these species kind of cluster together. There's no definition for a genus, which is why I'm a little bit iffy on the whole subgenus thing. They should just be genera. But with subspecies, the reason that I can that I'm not entirely sold on the idea is because there have been so many different definitions for a subspecies over the years. In the sense that, like, you know, like this is actually something I wouldn't mind asking Rob about with the Oreocryptophis, because I don't know anything about them, but they're all classified as different subspecies, and I'm just wondering, is that a data deficient thing? Have the genes been done on that, or is it kind of like because that's one example where I can't say, like with the carpet pythons, I can see, decline, so to speak, from, the southeastern up to the Cape York Peninsula, and then there's kind of the break in the Gulf of Carpentaria, and then you've got the Darwins and whatnot. But I don't know anything about Porphyes, so I'm actually kind of curious if you know much about that.
Yeah, so I mean, a lot of the Dr. Kevin Messenger's book, the rat snakes of China, is really good and has a great. Dr. J loves the pinpoint location stuff, and his book utilizes that in terms of the structure across the Porphy subspecies forms. This actually leads right into sort of my main thought or point, right? Across this argument, and kind of fit into both, and the Oreocryptophis is a perfect point. So, before I get into that, short answer to your question, there is a lot of discrepancy in available data, just because it's such a broad range of area, that I would say the short answer is yes. There's definitely not, it's not like there's a clear line of records in the way that we see with carpets in Australia to see that range. What I would say is that one utility of taxonomy or scientific nomenclature, right, is the ability to ensure that we're all talking about the same thing. And I sort of take subspecies as being equivalent to form. So a recognizable characteristics that the average person can, non-lay person or what, with some expertise can look at and perceive, oh, that's different from X is different from Y, even if they're fundamentally not that distinct, again, to Justin's point, relative to the sort of, depending on what taxonomists we're talking about, relative to their degrees of separation or years of separation and all those things, right? So to me, I think a lot of the utility in subspecies is really giving us a universal name for what are perceived to be different forms. And that's sort of, I guess, where I would see it fitting in. And that's probably what we really have with the Porphyes, is a situation where, okay, I guess the one that's actually, to me, and maybe again, it's a data deficient thing. So Porphy is from Porphyraceus is from India, and that, as a non-exporting country for the entirety of the time, those species really have been sort of aware. We've been aware of them in the hobby at least, but really we're talking very few specimens of those overall. In some ways, I guess if that's the nominate form, it looks closest to between a pulchra and a laticincta in terms of being a heavily banded snake, but without the laticincta's characteristic of orange into red. But all of those, right, are, I guess they're called, I also see that as a point for you, right, relative to saying structurally they're pretty similar. They do have size differentiation. There is sort of how they align along the spine, right, whether they're kind of bread loaf shaped versus circular. There are some differences across those forms, but those are probably, to me, species rather than all being, they all have to be this Porphyraceus from India. At the same time, it's serving the utility that it does in terms of saying they're certainly all different from one another, right? So that you do see this range. There is some sort of in-between specimens on a couple of them where we've gotten additional records in the last 5 to 10 years, as I say, that are Dr. Messenger actually put together a really cool PowerPoint on that. We had a conversation, I think, on colubrid and colubroid a handful of years ago, where or maybe it was even off the chat, and then I had gone on and talked about it a little bit, but he had a whole PowerPoint slide of all these different visualizations from different spots, and it was interesting that there certainly is regionality to those looks, but yeah, there were some interesting ones for sure.
That's kind of the complication too, is because you get into different habitats, and that's definitely going to push for different visual morphotypes and things like that. So relying on color or pattern or things like that, it can be deceiving, for sure.
Yeah.
And so I guess that's the kind of the coastal carpet, jungle carpet thing where,
Yeah.
if it's in a plain, it looks like a coastal. If it gets pushed in the jungle, the black and yellow does a better job of hiding them from predators or whatever. So, that's those kind of pressures I imagine if you took a yellow, or a brown colored coastal and put it in the jungle in a protected, arena, eventually the babies would start looking like jungle carpets or something, if it had selective pressure from predators and such. But other than that, I guess we have a lot of species definitions too. And so I've seen different taxonomists use different species concepts. And so, I think that's kind of what's driving some of us insane, especially, like, why did you go with that method? Or, the folks that just did just did
right.
a few years ago.
Well, yeah.
Yeah.
It's funny you mentioned. So I went herping with somebody who's actually worked in Damien Esquer's lab.
Yeah.
And so I got to talk to him a little bit about it.
Oh, cool.
And it was, well, so actually, sorry. So one thing I'll mention that I wanted to, that I've thought about before is with carpet pythons, that when we're looking at differences between animals based on morphology or based on scalation or whatever.
Yeah.
It's kind of like we're using those as a proxy for genetics. And so while I agree that like it's multifaceted, I think that at the end of the day, assuming that you have enough genetic data, that's the caveat, is that assuming that you have, like, you can't be using just 2 mitochondrial genes to separate species.
Yeah.
But assuming that you have enough genetic data, theoretically, that should be the species concept. Now, there's nuance, like 2 species can exist with some level of gene flow and whatnot. Like there's lots of caveats, and that gets really complicated when you get into like continental taxa.
Right.
But with carpet pythons, the thing that I've thought about a lot is that they are such a polymorphic species that they are especially susceptible to being selected for in relatively few generations for whichever habitat they're in. And so, the thing that I hear a lot about in captivity in herpetoculture is people say, well, why haven't people taken ball pythons like the carpet python people have and selectively bred them to make prettier ball pythons? Like, everybody's just focused on these multi-gene combos. And I would argue that that ball pythons just aren't that polymorphic naturally.
Yeah.
And so inherently there's the natural polymorphism in carpet pythons make them a great candidate for those adaptive pressures. And so, I don't know, like, talking about the southern coastals and the diamonds, I don't know if in a perfect world where I could see all of evolutionary history and I could see, okay, these branched off from here and they made a gradual climb up north, and like I could see a world where those are the same species. But in the same vein, if they separated at some point, there was some barrier to gene flow between the two, and then they reconnected where we now have integrades. Yeah, there is that level of gene flow, but they could theoretically be 2 separate species. And that's where I think the genetics don't lie. As far as I know, again, I'm a second year undergrad. So,
well, we like to pretend we're, keen on taxonomy as well.
Yeah.
We're I'm sure any taxonomist listening to that is probably like pulling their hair out going, these guys are morons. But
right.
Definitely.
Yeah.
But I mean, just kind of logically thinking about things, I guess, a good example is kind of that split between Southern New Guinea and Northern Australia, where you don't really see that much difference. They were connected, 8,000 years ago in the Pleistocene. And, we love the Pleistocene, of course. But, so there was some gene flow at that time, but now there's water there and there's no gene flow. And so, since it's so dynamic and it's changing all the time, is that really a different thing, or is it not, And so I guess I could go either way on that, but I guess the idea is you recognize there is a barrier there right now. And so
Yeah.
however we're looking at them, they're changing on their own evolutionary trajectory for the moment. Now, is that change going to be far enough along that when they recombine or have that access again to that gene flow across the Gulf of Carpenter or the straits there, is that going to be, are they going to say, wait, I'm not the same species as you? Or are they going to go, oh, finally, my long lost lover across the sea, here we go again, kind of thing. So, and of course, you see genetic drift even so I love the example of these. So we do research with mice, and we get mice from different breeding facilities for our studies with virology. We got mice from one facility, and they just wouldn't replicate the disease parameters that we were seeing in the same strain, inbred mouse strain, that was from the same company, but at a different location. And for whatever reason, those mice were resilient to whatever virus we were trying to infect them with. So, the mice from one facility would die and the mice from the other would not. And so there was enough genetic drift between those two populations of the same inbred mouse strain that they were different enough in regards to virus infection. Does that mean they're different species? Probably not, but we should recognize that there's this form and there's that form, or else we're going to run into trouble in our research, if we get it from the wrong, we try to use the wrong form for the wrong study. So if that, kind of illustrates a point, there's got to be something to that, And Ben would always kind of mess with people and say, once you take it out of the wild, it's on a separate evolutionary trajectory. So therefore, it doesn't belong to that species. It's just a snake in a box. So he'd always mess with people who are purists, trying to keep everything, all the lineage pure or whatever. He's like, nope, sorry, you're dictating who it breeds to. So therefore, it's not that species anymore.
And I believe that is a species concept, right?
Yeah.
Some people will argue. Yeah. Mm-hmm. And so right. And that's the perfect example of that's the issue, is we can't see into the future where theoretically those New Guinea carpet pythons could be incipient species and they could be on their own evolutionary trajectory. But then, what if we have an ice age and all of a sudden they're reconnected? I would say based on the genes based off of what I've seen and based on the genes as they stand currently, they're probably the same species as Cape Yorks. But who knows? A million years from now, they could be whatever. And, maybe carpet pythons being so adaptable, they might be able to evolve faster than another, say, I don't know, say your ball pythons. Like if a ball python got stranded on an island somewhere, it probably is going to stay a ball python for a lot longer because they seem less adept or less able to adapt to less polymorphic, and inherently a more I think the Chianactis, the sister group to Chianactis, the Sonora, are a great example of an incredibly polymorphic taxa, to the point where you get ridiculous amounts of within population. Some are banded, some are striped, some are unicolor, some are brown, some all over the place. And so they actually make for almost an extreme example of that potential adaptive radiation that can happen.
Yeah.
But yeah, so that's why I'm not sure if you I listen to a lot of podcasts, and I've heard you talk about the Herp Highlights folks, but I don't know if you know about the Squamates podcast with Mark Schertz.
Yeah, that's a good one too. Yeah,
it is. I wish they would do more of them because I really like that one.
Right.
But he's talked about, and because he is a taxonomist and he is somebody who's very, by necessity working in Madagascar. And his species concept is the unified, his species concept.
Yeah.
The one he chooses to align with is the unified species concept, where, every species is kind of evaluated on in its own frame, without, we're not trying to put things in boxes necessarily, but we're treating each species like its own hypothesis that needs different lines of evidence to be drawn in order to support it. So I don't know where I was going with that, but yeah.
No, I mean, I think that's the way I kind of see it as well, is you use all these you use all these different tools to say, okay, there's a reason that these are a different form than this, and you can bring in genetics or isolation from others and or morphogenics or whatever you want to use to say, yeah, these are a different form or a different type or whatever. And however you want to classify that is fine with me, as long as you recognize that distinction and that difference. Obviously, like the Kimberley rock monitors from the Kakadu are completely different from those in Kununurra.
Yeah.
I mean, they're separated by hundreds of miles of no man land kind of thing, guaranteed they're different species, at least, So I think saying, oh no, they're the same form, that's just silliness. It's just insufficient data. So we just kind of go, well, they look the same, so they're the same kind of attitude. Yeah. And they don't even really look the same either. They're very unique in the way they look. If you saw pictures, you could probably pick them out if you'd seen enough. So I guess maybe it's more impatience on my part. Let's get this figured out. But, the fact that they have to be funded and they have to go out and collect samples, that stuff takes time. And then figuring out what to look at with genetic analysis, what's going to change and what's going to be hypervariable and what's going to, be more stable and structured and, using all that. We're still kind of maybe not so new on the scene with genetic, use of genetics for taxonomy, but I don't know. That's the struggle, I guess, that I'm having is just impatience. Like, come on, we know there's something different. Let's get it out there. Let's make this known, and not the hoser method of saying, like, I'm just going to name everything and then let somebody else do the work. So yeah, that's the trick, I guess, with taxonomy. It's just a frustrating thing.
Yeah. Another recent example that, I think I'm not quite sure how it fits in here, but I do think it's of note is, so as part of putting together my forms lists relative to U.S. venomous and whatever, I was reading through right and right. A lot of the utility associated with old literature to me is looking at those things that were called subspecies before that we don't recognize now and saying, okay, are there defining characteristics here that would fit into calling this a form, right? So that would give us Atrachodatus. But an example is actually in the coral snake, so in Fulvius. As I was looking through that, I was previously unfamiliar with the postulated form, essentially the Dade County coral snake, right? So all of Fulvius from Louisiana up through North Carolina was considered Fulvius fulvius. And then in 1928, Schmidt, based on a specimen from 1920, had said, oh, well, actually, these ones in Dade County, they have reduced black stippling in the red bands. And in some of the males, there is not a divided supraanal scale, and it's like, okay, I don't think I have to actually find one in Dade County. I don't think I'm going to put that as a form on my list, man.
Yeah. Less black speckling.
It's funny.
And they did highlight, or highlighted in the description was saying, well, we see the same thing in the Getchula from that area. Essentially, the hypomelanism that's sort of common to animals in that area. And I believe Dade County also includes the Keys, right? So if you're talking about Keys corns and I just said the Getchula, that's in the description. But I mean, nevertheless, right? If we're saying this is something that's taken as, acceptable to have, it's all one form from far eastern Louisiana up through southern North Carolina. But this one county, and we're getting reduced black stippling in the red. I don't think even my sort of comprehensive nature would take that into the list.
Yeah. It's funny too. So similarly in my research with the Sonora related stuff, there's over 100 years of literature classifying and reclassifying Sonora because there's these different morphs. So I would almost argue, so and subspecies, like what you were saying before, it is almost just like an impatience thing a lot of the time, because now we can recognize that all of these things, well, there's two different clades, but more or less all these things are the same. They all interbreed, and they all, they all evolved from tricolored ancestors. And then it kind of seems like they're losing that because a lot of the populations no longer occur with a species to mimic. But, there used to be Sonora miniata, and then between those, there was miniata, and miniata linearis to denote the difference between the banded ones and the striped ones. And it's like, and now we understand, there is population difference, but the difference isn't in the phenotype necessarily in like what they look like, but it's in the population composition and the proportion of the different phenotypes. And I mean, there's so many more. There's Sonora saxatilis and Sonora erythrophis, and like literally so many that are no longer used. And so, yeah, and it seems like a lot of the time subspecies are used kind of as a way of saying something is something without necessarily having the data to back it. And it's used as a placeholder. And, but that's one of the uses. Like, and then, you know, then there's the whole, like the Ensatina debate with, you know, it's
Green
used
species.
to split up a cline.
Yeah.
And so I think that the issue isn't with, I agree that I think that splitting up species into distinct groups to make it easier for the sake of being able to talk about them, you know, that's why we have taxonomy, is so that we can talk about animals in a way that everybody understands. But at the same time, you get a lot of confusion when the definition of subspecies changes from person to person. And I think that it's because subspecies are such an maybe not unregulated, but not as thought about, say, as the species definition, it creates a lot more chaos.
For sure. We got a suggestion a while back from Mark Heath to talk about using taxonomy as a tool for conservation. And I wonder sometimes, like, I wanted to bring up an example and get your thoughts on this one. So the Shark Bay Woma population, where they've been basically extirpated, you know, throughout the wheat belt and throughout a large swath of Western Australia where they used to occur. We haven't seen records since the 50s or 60s. And they were pretty few and far between at that time too. So, this little island, you know, population of womas that have persisted in Shark Bay, you know, do we classify those as a different subspecies? Because they don't have genetic flow with other populations, and it would have the added effect of recognizing them as something distinct and worth protection. What do you think?
Well, and so just real quick, so there's a topic, again, as a forms herper, my question has been, do I have to find cryptic species, right? Exactly on the Ensatina context, or in this context, right? So if the Woma that I saw was in Shark Bay, have I seen a Woma? And that's really sort of the inverted angle of that question, you know, and not so much in the subspecies discussion, but I've been playing with that idea a lot, and my feeling is generally no. So to me, rather than Nipper's approach, right, where he's trying to find and I guess he's finding species, not necessarily subspecies. I think he's found all but maybe a couple of the subspecies of snake in Europe, right? But in terms of, okay, if we're talking about cryptic speciation within a population, right? If you've seen one at that area, have you seen them all? If you can't even know, right? If the only determination is available based on a non in hand look, right? It's all on the inside. What do you do with that? And to me, they have to be the same, because if you can't tell them apart visually, then you know you've seen the form from that place. Yeah.
Well, and that's I think the difference. Oh, sorry. Go ahead.
Nick. No.
Oh, I was just going to say that's, I think, the difference between like a herpetoculturist, a herper, and a scientist in this regard is, like, I'd agree with you. Like, if I, you know, hearing people talk about frog taxonomies, it's. I mean, I do not disagree with, like, you know, I've heard a couple people talk about how the validity of using bioharmonics to differentiate species. And I don't disagree with, you know, if the genes don't lie and there is a way to differentiate them, even if it's not physically, I think that that's a valid taxon. I don't disagree with the idea or the concept of cryptic diversity. But you know, being a field herper, you know, I'm currently, while I'm home, trying to find every subspecies of Ensatina just for fun, because I'm currently unemployed and just have gotten into photography. So, but I'm not also trying to find all the intergrades, because that's, a task in and of itself. Like I've gone around the top of their range and found Sierras and Oregons and Yellow-Eyeds, and I found a couple of intergrades by default. But it's like, I would say that a Sierra integrated with an Oregon it looks, you can tell what it is. You can tell it's a different thing, but is that a different thing?
Yeah.
And that's where ensetinas are a little bit of a weird thing, because they're not like really a ring species. Like they're kind of they're used as that example a lot of the time, but there have been barriers separating their range over time. So, but I agree with you. Like, yeah, if I was herping for them, I'm not trying to find every single cryptic species.
Well, I mean, I guess you could go back to the Kimberley Rock Monitor example.
Yeah.
Would you want to see both the Kakadu form and the Eastern form, with the idea that they'll probably eventually be split?
Yeah.
And we'll have two different species there. So you might as well, if you're in the area, you might as well try to knock them out while you're there, kind of thing.
For sure. But that's a much longer period of separation, right? So with the shark base stuff, we're talking maybe, and I get your point, right? Relative to future state, being in this isolated pocket, what might happen. But we're talking about, well, there was a band of, of range up to 50, 60 years ago, something like it's in the last certainly in the last couple hundred years that we've isolated them based on human activity, right? Human habitat modification. I was just thinking too, right, in that same vein, something Justin and I have seen, right? There's this particular spot in not quite in the boot heel of New Mexico, but where you get Mojave prairies, right? And they're very, have a look, right, to them. And we've seen one, right? A little one.
Yeah.
And it's like, well, is that a thing? Well, they certainly are recognizable, as you say about the ensetinas, and they're known from that spot, and not really, just, it's not a general thing. It's something particular to that location. Is that then a form? Does that go in the list? I don't know. And I've kind of said no, even though it falls into that, okay, it occurs only in this particular place.
Well, it'd be like the intergrade zone for, coastal carpets and diamonds. If that form is a different form.
Mojaves are prairies,
That's just a mix between two different forms that are unique and identifiable. And then when you have the mix mash, it's a little
also
less easy. Yeah. It's like, you can still tell that they're a mix, but that was interesting seeing some of those crossed crotalids too, like the clobber mixed with the willardi, was it clobber and or lepidus willardi hybrid? Kind of cool. But
and an interesting example that I'd say is what was formerly the two subspecies of Varanus storai, where to your point about field herping, if as they were subspecies, I'll admit I've never kept either, so I can't tell the difference visually. I'm sure if I spent some time looking at them, I probably could, but they're relatively similar, and they're not necessarily high up on my bucket list to see. So if I ticked one, I probably would have ticked both boxes. But come to find out, storai are more closely related to the Acanthurus complex than they are to Acreatus. So that's an example, I think, of where taxonomy might influence my field herping, where I'm now like, well, if this is clades with this, and I would definitely consider Acanthurus to be way different from Acreatus, then, and that was the basis on a broader scale, why, going back to the Chianactis, that was the basis for which Chianactis and Chilomeniscus were sunk into Sonora, is because it was found that Sonora were paraphyletic. So you bring up a good point, and that's where I think that like hobby taxonomy almost needs to be kind of its own thing. And we need to stop arguing, like when Esquire sunk what was it, Stimsons into children's pythons. He wasn't saying, all right, everybody go breed your children's to your Stimsons pythons. He was just saying, hey, like genetically this is how it turns out. And I've looked at it, and I don't know, it looks okay to me, but I'm also not gonna, if I get children's and Stimsons down the road, they look different in the hobby. I don't know.
Yeah. Well, I mean, and I think they showed like data that supported the opposite too, where it looked like there was pretty distinct.
Yeah.
They said, well, this one came close to the range, so we can't say there's no interbreeding, but you can have interbreeding between different species.
Well, what I saw, I read it a while ago, but the way I saw it is they had, the way that it played it out is the currently recognized children's pythons and Stimson's pythons are not what they are genetically. And so we have this population of children's pythons that would be children's pythons, but they aren't associated with the children's pythons that are currently children's pythons. And so it was something like, we can either split these into 3 species, but that doesn't really make a whole lot of sense. So let's just sync them for the sake of not, not changing the definition of what a children's python is, or resurrecting a new species name, or not resurrecting, but naming a new species, or whatever it was they chose. Again, I'm not a taxonomist though, so I don't know.
Yeah, it rubbed me the wrong way. I think they should have gone the opposite way and separated those.
And that's absolutely fair. And that's why, the same deal with Sonora. There's a paper in the paper that sunk Chylomeniscus and Chianactis. I'm like, you had the choice of either resurrecting, or of either naming 2 new genera, or sinking all of them. And I'm like, you should have named the 2 new genera, because you elected to use subgenera instead, which is its own can of worms.
Yeah, for sure. And, I guess that's just the thing, is we have an idea of what they are, especially hobbyists, that mentality.
Yeah.
I agree. Yeah. We do have kind of our own taxonomy.
I mean, we have localities that don't follow, scientific, look at the Alterna, the Alterna localities. It's like, is it really that much of a stretch for us to just kind of have our own, hobby accepted taxonomy?
Yeah. Well, and another funny thing with that was, we had this idea of what a carpet python looked like based on what carpets were kept in captivity in the U.S. And so
right.
Said, this is a jungle. This is a coastal. And then I went over there. I'm like, this jungle looks like a coastal, and this coastal, it's like, there's no, this coastal looks just like a jungle down in southern, near Brisbane, and it's a very jungly looking animal. And so, I mean, there's so much diversity in phenotype. And that was kind of one of the ideas behind the first book, is to, lay that out, and show that, illustrate that. So, I hope we accomplish that, but, and I think too is like Rob was saying with the old books from the 50s and 60s and stuff. They were pretty data deficient back then, especially in regards to reptiles and their distribution. Now we have so many people going so many places, and cars and trucks and running over stuff, and so we have a lot more records and a lot more idea of what happens where, to some extent. Of course, there's still some gaps in our knowledge and holes. I still want to go looking for those Brettles pythons that are up northwest of the main population, kind of isolated out there. I think that'd be cool to go. Somebody needs to go find those again, because I think there's like several records from
the Narrows, something like that.
The granites,
the granites, yeah.
So why nobody has gone back? Well, maybe somebody has, then they just haven't published that. But that would be kind of a fun little expedition to go up there and you could go to some really cool habitat. I mean, it's out in the middle of nowhere, but you could get some cool WOMA activity out there, in that area too. But so that we have this idea of what things should be based on what we want them to be, or what we think they should be, and whether or not that matches with reality. I'm not suggesting there should be a separate Brettle subspecies, but maybe the line, is, and I guess one example is somebody just recently, or, within the last 5 or 6 years, found, Amiae, Nephurus, Amiae in like over the border in Western Australia, kind of outside the accepted range. And, that's kind of cool when you see those things pop up. Now, of course, somebody just found a blackhead down in, the Null Arbor Plain. And I don't necessarily think that is a natural occurrence. Somebody probably let it go, or it escaped or something out there. But or hitched a ride in a truck and ended up in the Null Arbor, so I don't think we should get excited and have a range extension. But, those
and talking about your carpet pythons, like, Australian herpetoculture could really throw a wrench in a lot of those things, because all of a sudden, coastal carpets are showing up in Sydney and vice versa. And I'm not saying it happens all the time, but there's definitely, I'm on a couple of Facebook groups where people are like, we'll post a picture and say, hey, I've got this snake in my yard. Can someone come get it? And they're like, oh, that's a jag.
Yeah.
That's not supposed to be there.
Right. Or a Brettles or a
Brettles or whatever.
I think there was an olive python that was weirdly misplaced. And,
And you see those records from time to time, like, that there's this or that way outside where it should be. And is most likely an escaped captive or a hitchhiker,
Yeah.
I mean, sometimes those road trains go long distances, and I could easily see one just kind of curling up somewhere up in the inner workings of the truck, and then falls out thousands of miles from where it should be. So, I don't know.
The thing with that blackhead that's interesting is a question of like how long. Okay, presuming it wasn't, it's not shenanigans on the part of the poster. Yeah. Is how long do you think that could survive there? Right, as you highlighted, it's well below sort of the latitude, right, that they're
adapted to be in.
Right. So the question is like, and maybe that's in a different year, a different time of year, maybe it's a different answer, but it's an interesting question.
Well, and I think that's well as illustrated by the Burmese pythons in South Florida. When do they become their own taxonomy? I mean, they're definitely having different environmental pressures and cold snaps and all sorts of stuff that they're adapting to.
Well, haven't they proposed? Like some people have proposed naming them their own thing, and
Floridensis or something.
Yeah.
Should they?
But that's another example of where like we can't really see the future. Where that's a pretty obvious example of like, hey, these things are never going to come in contact with native Burms again. But I think the whole Australopapuan region is a really interesting case of it's evolution in action, right? Like, we can say these, Papuan carpets are different from Cape Yorks and Papuan, I don't know, scrub pythons or frilled dragons are different from Australian frilled dragons, but at the same time, are they different enough?
Yeah.
And that's where the nuance comes in. I did want to, because you mentioned the potentially using subspecies or taxonomy as a tool for conservation. And this is something that I've thought about too, because one of the exceptions that I've thought about before to when to use subspecies is like, hey, this is, subspecies are recognized by conservation organizations and by governments and are valid. Like they can be conserved. I mean, look at the San Francisco garter snake. Like you can't touch them, but a red-sided garter snake is, fair game. And so I've heard that argument before, but I just this might be my own personal opinion, but I just think that it's doing a disservice to the science of taxonomy to kind of use it as a tool where why can't and this is more of like a question for, governing bodies and whatnot, but like, why can't a population, your Shark Bay Womas or locally we have a Tule elk reserve that is isolated to the point of the tip of Point Reyes, that is the same subspecies as all the other Tule elk, but they are protected just like the other Tule elk, the individual populations. Why does it need to be its own subspecies to be considered a population that's worth conserving? Because then you would be kind of making an exception to the rules of taxonomy in order to do that.
Yeah, no, that's a good point. And I think may confuse the matter, because if you ever wanted to reintroduce some animals into that historic range, like they have tried to do with certain things. Yeah. Indigo.
Yeah.
What if they're considered different subspecies or something, then all of a sudden you're making all these natural or intergrades or something, which is probably not the case,
Right. Well, I mean, tiger quills are a good example of that, where I believe I'm not sure of the exact species, but they've recently released Western quills into the Flinders. I'm not sure if it's Westerns or tigers or whichever, but essentially the original population, whether it was a species or subspecies, was extirpated. And now they've brought in Western quills, which are notably not the same thing as what once was there. but they've evaluated the area and they've determined that they make they've been able to make an argument for why they should be introduced into that protected habitat.
Yeah.
So again, it's not really the taxonomy. And this is where I guess the difference is between a taxonomist and an ecologist is like an ecologist just sees if it's fulfilling that role in the ecosystem, it's the same thing.
Yeah.
But a taxonomist,
Interesting.
It's super interesting you bring that up. Jordan just mentioned it. I knew that was what you were going to say, Justin.
Yeah.
Jordan just mentioned that last week. So.
Oh yeah,
he saw a few of them in the Flinders. So it's a successful reintroduction.
yeah. I mean, and they're talking about them like competing with cats and stuff because they're that badass. Maybe not in the Flinders, but elsewhere. It's funny you actually mentioned Jordan because he's I met him at IHS.
Oh
yeah, and he actually
spoke highly of you. I can't remember if that was on the recorded podcast or if that was just afterwards, but he mentioned you. He's like, "Oh yeah, Nick is a great guy." So
I know.
You just missed each other in South.
I know it was a bummer that didn't work out, but he was a huge help. I mean, even in when I went and found my Kyanactus, he was I was on the phone with him and he was coaching me through.
How
to find him and all that. So.
He knows his stuff when it comes to Southern California, man.
Seriously, I'm looking forward to hearing his episode because when he sent me the picture of what he found, it's pretty spectacular.
Yeah, that story was probably one of my favorites from that episode. Just it hits home, I mean, I definitely wish we would have found it when we were there, but it's really cool that at least one of us found it.
No, definitely. I mean, I didn't see any snakes when I wasn't in that area, but I didn't see any snakes while I was down there. And
it's very snake. It seems really snake deficient.
It's interesting.
I mean, there's records of Stimson's pythons down there, but they're not very. They're kind of few and far between, and the carpets are hit and miss, and there's some womas a little further north that we went up and tried to find, and we saw a track that looked like it could have been either a really big mulga or a woma. It was a big snake, and so we just missed one maybe. So that's when you have something that spends 70% of its time in burrows or underground or more, it's hard to catch those out at the right time. So. But that's same kind of thing. We're like, where's all the snakes? So I mean, we saw Suda and a big mulga, and that was about it on our trip. We saw 2 snakes over 2 weeks.
We tried. We put in I was only there for a couple of days, but we definitely put in the hours. And I kind of wanted to see a Brachiophis just to be able to just to compare it to our American shovel noses. They're very convergent, and. Yeah, we did. We saw a dead Western brown. I think we saw a DOR Western brown, but that was it. Yeah,
it's pretty crazy. Speaking of those Brachiophis, I've seen a couple different species of those, and I mean they're quite a bit larger, of course, and they seem different. Like they kind of have a different structure to them and stuff. So it never dawned on me that they were convergent with and Kyanactus. That's cool.
Yeah, and I am very vocal about my love for Kyanactus and Sonora and all of them. And so I posted, there's a Facebook group, and Jordan actually commented and was like, and mentioned how they're behaviorally quite different, and how your Kyanactus are a lot more flaily. But so that's why I wanted to find one, right? Because they are they're called. They're called coral snakes, I think sometimes. And because of that banding that they can have, and it's just interesting that they're not only sand swimmers, they not only have that same rostral adaptation, but they also have, some of them are unbanded, some of them are banded. Whether or not the modes of inheritance are the same, they're remarkably convergent for being, not even colubrids.
Yeah, that's a good point. That's cool. It is fun to see those examples. I'm looking forward to hopefully seeing some more thorny devils. We did get a thorny devil. Did you see one down in South Australia?
We tried.
Yeah.
Did not. Surprised a week later he sent me a photo of one. Because that's how it goes. But no, we didn't, unfortunately.
We got one, and that was pretty awesome. I think that was Jordan's first thorny devil. I'd seen them in Western Australia, and I'd seen a few DORs in Alice Springs.
That's how I've heard a lot of people see them that way, where they're just so slow going across the road that like most people, they see a couple DORs before they see a live one.
Mm-hmm. And the ones I saw in Western Australia, we were going probably 60 miles an hour, and I saw it on the road, kind of like, wait, that was a thorny devil. Flip around and get it off the road quick before a road train took it out, but I don't think we saw any DORs in Western Australia, and
that's good.
So I don't know, maybe there's not enough people driving down there. That's the other beautiful thing about Western Australia is it's pretty, get out there. There's not many people out there. And so, it's pretty cool. I'd highly recommend it. Rob almost made it over there. We were going to go on a trip out there, but then COVID came along and ruined that plan. So we got to reschedule that trip, I think. But too many cool places to see for sure. But
it's too big of a country. Every time I try to rank the different places I want to go to, I'm like, oh, but the Kimberley. Oh, but I really want to, being a monitor enthusiast, I'm like, I've got to get up to the top end, and then, oh, but Western Australia. Oh, but Cape York. Like, it's just too many.
For sure. Yeah. Well, I think on this last trip in Darwin area and then over to Western Australia, we got Mitchellii, Mertens, Scalaris. What else do we see? We saw, I saw Ocreatus, Flowered Eye.
That's the one. Pete saw Glebopalma.
Glebopalma. We missed that. We missed that. I was really bummed out about that because we spent a lot of time looking for those things and just sitting in the same place, staring at the same patch of rock. Trying to recreate what had been seen 2 days in a row. So what do you do? And we spent a lot of time in their habitat, but man, I saw tracks of one, I'm pretty sure, because it was right where a number of records were, and I went through, didn't see the tracks, came back, they were there, and right by where records have been made.
I knew we wanted to see them.
They're so cryptic, aren't they?
Yeah, they're just so shy and fast, and they do a good job of staying out of sight when you are around them. So.
Yeah. Well, and even the one that Keith saw, I think he saw it for a grand total of about 8 seconds across 2 days.
Right.
So, And even the difference between Mitchellii and the Scalaris, at the spot where they co-occur, the Scalaris were very bold, relatively bold, for sure. The Mitchellii were all incredibly wary, and that was almost a defining feature of, is it a Scalaris or a Mitchellii? How wary was it? Yeah.
Quick. Yeah.
Well, I mean, one of my, pretty high up on my list, up in the Cape York Peninsula, is I really want to see a Dorianus up there. Mm-hmm. And that's like a prime example of,
spend weeks and you might get a glimpse of one, right?
I mean, I know people through the AHS who have spent a lot of time up there and never seen one.
Yeah. I think I'd rather see one of those chlorostigma or whatever is that what they're called? The indicus type? Those things are awesome looking.
The speckling on them is pretty unreal.
Yeah.
Well, and given Australian sort of politics and realities and things, my question, if we went back 20, 25 years, my question was, well, do they actually exist there? Is that just to legitimize sort of the captive animals that they, sort of, no, this is from New York. And so that honestly clouds a lot of those questions. And then even gets into, Becarii versus Keithorni and, these, you saw indications of a Keithorni, right?
No, we saw
a dead one.
Yeah, I said DOR and a live one. I heard kind of a scuttling on the side of the road, and so I just kind of chased after that sound, and then I saw it climbing up a tree, and then it went to the opposite side of the tree. So I was trying to get a picture of it. So I'm circling the tree, trying to get a glimpse of it. And my dad was there, and he was on the road still. And Rico Walder was with him too, and they were watching this thing go up around, corkscrew the tree. So they got to see it several times. But a lot. I did not get to see it very well. I got glimpses of it, as it ran away. But we got a really good look at the DOR. He didn't run away too much. It was kind of sad to see, somebody had hit such a cool little monitor, but neat speed. And it was actually a lot bigger than I expected. It was, kind of a larger than I would have thought for a tree monitor, but very, very interesting too. Like the diversity of the tree monitors too, watching that, video, with, oh my gosh, now it's gonna leave my head, of course, as soon as I start talking about it. But on the Trap Talk Network, the new monitor.
Oh, Chris Haplin.
Sorry,
Yeah.
Chris. But yeah, he's a cool guy. But his podcast is real. I really enjoy listening to that. But
I think I have to watch it on YouTube though, right? Because he has a lot of visualization. At least the one I was just listening to, it was like all sort of references to these pictures. I'm going, well, this kind of doesn't work as
not a great podcast. Audio media.
Yeah. I think, the tree monitors are kind of a really cool example of, I think the green trees and his argument that they kind of parallel the chondros and their different lineages. I think that's pretty interesting. And I mean, it makes sense. I guess it's just hard with New Guinea being the way it is.
Yeah. Well, I think that brings up that, speaking of green tree pythons, kind of the idea of island, when they hit an oceanic island rather than an offshoot or a peninsula that's underwater kind of thing. When do they start becoming another species or
speciation? Yeah, because of exactly.
And that's probably where we have to evaluate it as a freeze time, this snapshot in time, because realistically that's where we are, right? Like we exist on such a minuscule time versus the vast time scale of evolution that we kind of have to evaluate things as they are and not as they might be, because otherwise we'd go crazy trying to speculate about, are these, I mean, not to bring it back to Kyanactus, but there's two different species of Kyanactus, and I don't buy it. I think the paper that separated Annulata from Occipitalis is, I think that it could be. I think it's possible. I think that they didn't have enough data to support their argument. And the only geographical barrier is the San Bernardino Mountains. But there is gene flow. There's gene flow through the Coachella Valley. So, where do you draw that line? And I think this is an example of hey, these might be species one day, but I don't think they are right now. But yeah, you're absolutely right. And a lot of them, this is a pretty interesting thing with monitors in Indonesia is a lot of them have been split based off of trade specimens. Wolfgang Boma is the guy who did all that, and there's been some, a lot of what he's done has been valid, but there have also been, specifically in the Kai Islands, there's a bit of this might not be the way it is because these trade specimens might not be what you think they are. There's some type localities that are a bit iffy. And there's a lot of cryptic diversity there, the Indicus complex. So that's where it gets really confusing.
Right. Yeah.
Absolutely.
I lost track of that whole argument. I think that's why my interest kind of stops when you get to Northern Australia. I just Australia is a little easier.
Is such a mess.
Oh it's crazy.
It's cool though.
Yeah. There's some neat stuff there. I definitely love to check that out someday, but maybe after I've exhausted my Australia trips and I have a
right.
And I definitely wouldn't try and list or form herp it. It would just be all the cool stuff.
I think there's been some argument that Varanus finchi might occur on Australia, and then there's again, going down the rabbit hole, I've spoken to some people and they're it's a mess.
Yeah,
but it's definitely thought provoking.
I guess in regards to that, those green trees got to Biak somehow, and that, giving an example of an oceanic island. And it is conceivable that maybe some raft back to the mainland and more raft from the mainland to New Guinea. So how often do you need interjections of, that?
Well, that's where it gets, and that's the thing I find really interesting and that I didn't kind of think about until recently is that two different species can exist as separate entities. While still having some level of gene flow.
Sure.
And that's kind of where I'm not educated enough to be able to have an opinion one way or the other for a lot of those cases. But that's where I think the nuance comes in is a lot of people have this idea that, if a species needs to be genetically isolated, but if like you say, if you think that maybe diamonds are a separate thing from other carpet pythons, they do interbreed with coastals. There is an intergrade zone. So is that the case, or do they form a natural climb? Like, who,
Yeah.
it would be really nice to be able to see back in time to see what they did,
Right. Well, and I guess that's the trick, and I think it all comes down to the communication, right? If I think there was some politics involved with why they didn't give Biak a name, or either a species or a subspecies designation. But I mean, we should have, obviously, they might be the same species as the stuff on the mainland, but they're a different form. They're isolated. They're genetically on their own, distinct evolutionary trajectory. So, they probably should be a different species or subspecies or whatever you want to call them, but we should communicate that way, Biak is a different thing. So, Biak green trees should probably have some kind of designation, just so we can communicate and know what we're talking about,
And that's what I've, you hear a lot about certain people talking about how they named them as subspecies when really they should be their own species. And it's the case for more than just green pythons, but it's definitely, you're right. Like we need like a substrate to be able to talk about this. I don't know if that's the right word, but what I'm saying, a medium, to talk about these things. But I think that when you to your point, bringing in like a political decision, that's where I think it takes the objectivity out of taxonomy. And it takes, it all stems back to the question of whether or not taxonomy is a science, because if it truly is a science, then, you shouldn't be able to influence it with policy or with, potential conservation of certain populations. It should be an objective science. And a lot of people will argue that it is a science. But then some others will say it's too subjective. It's not, you don't have a testable, I'm not sure what the exact argument I heard was, but you get what I'm saying.
Yeah. Yeah. And I maybe we call it a soft science.
Yeah. Right.
I mentioned that my daughter's studying psychology, and I'm like, oh, you're in the soft sciences. And she's like, what? Don't say that. That's not, that's not nice. I'm like, no, it's not a dig at it. It's just, it's not.
Yeah.
Like, well, what does that mean? Yeah.
Yeah.
So had a little fun discussion with her, but
yeah.
Sorry, Rob. You looked like you had something to say, but that's
no, absolutely. No, I think you're great. I was just going to poke. Yeah, it's about what's a soft science? It's crafting and supporting an argument, right? And people can have countervailing points and all those things, and it's just trying to persuade, right, based on the evidence that you have. So
and I think that can speaking of the alternate localities, as long as you know what they're talking about or why they're specifying a certain road junction in West Texas, when they're talking about their gray bands, it just helps you communicate, okay, I'm crazy. But
that's the best.
That's what you're communicating. No. But you're at least keeping things, in the structure that you would like to see them in. And I think, kudos to those people who kind of went to that effort and went to that level and has that, that specificity of, that it's kind of cool. And you go to different localities and you see maybe some nuanced looks and some different kind of looks to them. And you're that's cool. I like that, so why shouldn't that be something that we get excited about and celebrate and give a little name to, Yeah.
I don't know. Well, and especially for the hobby, right? Like, the hobby is, some people, the alternative being the extreme example, and maybe chondros being kind of the other end of the spectrum where people just kind of hybridize them together all willy nilly. You're right. It is cool. And don't get me wrong. I think it's up until recently, I thought that, all of the Kayaktis north of the San Bernardino Mountains didn't have red bands, and all the ones south did have red bands. But then I have seen a bunch of pictures now from the Anza Borrego area of like some really weird looking annulata.
Yeah.
And so now I want some. Now I want to go find them.
Yeah.
And so, I think that's what draws a lot of people in, right? And that's where I just think that that's where I just kind of have it in my head where I can compartmentalize my scientific taxonomic brain and my hobby, forms brain.
Yeah. And I think too, it just helps us to bide the time until, well, I don't know if we really do that very well in the hobby where, like you said, like look at the green tree pythons. It's just all willy nilly. Like let's try to make this weird designer looking thing. So we'll breed whatever to get that end result. And that's fine. I mean, it's snakes in boxes, right? They're on a different evolutionary trajectory. It doesn't matter what we breed them to in captivity. Yeah. We're not repopulating Biak or whatever. And and if you were repopulating Biak, you'd probably take specimens from near Biak on the mainland. Like replicate what probably originally happened, So I don't know. I think we get a little precious with it sometimes, and that's fine. We want to kind of preserve and I like to see something that's representative of its wild form or the wild type, so I can have
absolutely
slice of Australia in my living room or something, and not just have some mishmash of whatever. I want to kind of see the form that I saw in the wild, and sometimes that's really difficult because like I think Kimberley Rock Monitor is a nice example of that too, where they probably had different forms, different areas where they collected them from, and
you're going where I was going. I was going to say
Pilbarensis.
Well, that's yes, exactly. We're all on the same topic. Where I was going to say, in terms of hobby Pilbarensis in the United States, a little bit of discourse right on the Pilbarensis versus Hammersliensis, as something that perceived to be a singular entity, right, at the initial time of coming in, and then are subsequently split. It's the green tree thing all over again. It's the Kimberley, just saying, well, what did you actually have? What's over at Lucas's house?
Right. Yeah. Well, yeah. And I can say I have seen a lot of European animals that lean a lot more towards Hammersliensis than some of the, what we don't have with Australian species that we do have with some of the Indonesian species is locality information because they were obtained less than legally.
Yeah.
And so it's really anybody's guess of what they really are. Honestly, this is actually a conversation I had with Frizy. Maybe not the majority, but a good chunk of the Australian monitors we have in the US, because there is kind of a black box of information about where they came from, they're probably hybrids. Storai and could be with Ocreatus, Glaueridae could be with those other Glaueridae.
Well, and I got I got a Pilbarensis that had blue in it, and it was pretty clearly it had Glaueridae in the mix. So Pilbarensis that had Glaueridae.
Oh, weird.
Well, we know Frank did that crossing a few times.
Did he really?
Yeah, he bred Glaueridae to Pilbarensis a few times. I'm pretty sure. I know he had some hybrids back in the day.
Yeah. Or even, I mean, you could say I could go on Bushy and Gill and and all the others that we didn't know were different species. And so I can't remember where I heard it. It was on a podcast. I can't remember if it was maybe Dusty Rhodes or John Lasser or one of the colubrid, one of the Southwest colubrid people. We're talking about the motivation behind keeping localities pure isn't necessarily because you're right, because as soon as it's out of the wild, it's on its own trajectory and it's no longer representative of what that wild population is. But as long as you're keeping a locality pure, no matter what taxonomy does, you know that what you have is what you have is what you have.
Yeah.
And I can really,
Right. Unless you collect from, Dale's Gorge and then, a little bit further north.
Unless you're really getting on that
the razor's edge between the two different. Because I mean, Hammersliensis and Pilbarensis are not separated by very far. I think we saw whole species within a day or two of each other. It wasn't very far to go between their habitats. So, I guess we try to do our best, but sometimes we don't hit the mark. And for things like that, where it's, black market and, smuggling and stuff like that is involved. And I mean, I think a lot of that, you read Stolen World and you see that, they use a pseudonym, but we know, who was sending over Australian species and probably where he was getting them. He was either breeding them or collecting them locally or something like that.
Well, I can tell you, I know people who did collect. I know of people now who did collect those animals to be smuggled, and it's like it's not a. clarify, I know of people. I don't know them personally. But yeah,
I know
you're
some
right.
personally. Yeah, one of my friends got put in jail for a year.
Yeah, fair. Yeah.
Because he was bringing stuff home to fill in gaps that we didn't have. You know what I mean? Like,
right.
To make sure that we had the right, species, or we knew what we were talking about.
Yeah.
And unfortunately, they got confiscated and sent to the San Diego Zoo. And I went there. We went to see them in the zoo.
Yeah.
And they were on display, and they had all sorts of like Egernia that were later Egernia signitos, depressa.
Oh yeah. The depressa complex.
And they had them all in the same tank. And like you had the signitos over here and the depressa over here. They were like the warring gangs. And they're like, man, they're beating each other up. We don't know why they're so feisty. We're like, because you've got different species in the same cage. Before it was species. I mean, he recognized that they were different forms, and they were.
Oh, I mean, one of them is red and one of them is gray. Like,
right? Yeah. It's like,
and that's a very good example of like those are different. I mean, you don't need genetics to tell that those are different.
Yeah, they're isolated. They're completely different in appearance. And, it's like, and granted, I mean, hobbyists and herpers and herpetologists knew this for a long time. But again, it takes time to do that work. And so in the meantime, the zoo's like, they're all depressa. What are you making such a fuss about? Like, come on, guys, do better. It is really interesting that subspecific, not in the context of subspecies, but just like as a population level, like, and that's why, you hear it get thrown around a lot. For a while, people were saying, "Oh, I'm breeding." It was a common thing to say, "Oh, I'm breeding this species for conservation. I'm keeping it alive in captivity." Yeah. And then it was kind of a trend to say, "Oh, well, that's not actually conservation. Those animals are never going back to the wild." And it kind of it brings into question, like, where do you want to define the line of what is worthy of conservation? And in all reality, captive breeding or captive assurance colonies are like an absolute last resort of conservation. Mm-hmm. Like, this is it's not often successful. It's not often a valid, means of repopulating the wild. And so it's interesting to watch all that play out, I guess, and different people's perceptions of what conservation really is. Right. I guess if you found out there was some weird guy in outback Australia breeding thylacines together, it'd probably be okay for using those for reintroduction. Sure.
Like, and that's what I mean. Right. Absolutely. But then if you've got one, if your Biok, like Bioks are still on Biok, so there's no, nobody would argue that we should start putting Arus on Biak. Like, that's preposterous, or to the conservationists, it might be.
Yeah. Well, if you put Arus there, then you're mixing.
Then you're going to get some weird stuff. That's for sure. Yeah.
You get your
Rubiacs. Yeah, exactly.
We get all these we'll get designer chondros in the wild.
Yeah, exactly.
The sickness.
I don't know. Maybe that's somebody's dream.
They'll get picked off immediately.
Exactly. They're eaten immediately.
I know. I just imagine the sickness out there in the wild blending in with the undergrowth.
Yeah, right. They become a different species just because they look so weird.
Yeah.
But I mean,
makes me think of. Oh yeah, go ahead.
Oh, go ahead. No, after you.
Oh, I was just gonna say, it makes me think of the another convergent species, the basins, with what is it? Corallus. What's the other species? It's not Caninus. It's not.
No, the other one. Yeah, the other one.
It's the other one. The few pictures that are out there almost look like a designer chondro if it was an emerald. I don't know. Maybe that's some weird hybrid between. It's probably its own thing, but maybe that's some weird hybrid between Caninus and Betisi, and and that's what happens.
Could be.
We see Hortulanus and Caninus for sure. So like,
oh yeah, right. Yeah, and that one's what is that, like gray and. Yeah,
a whole host of different things, but they just don't survive really past a couple of years. It seems like they produce offspring, but not fertile offspring and things. But people have tried, but that seems to happen mostly, I think, at the sort of collection points rather than in the wild. But that's not, if it can happen at a collection spot, then you would think it could happen in the wild as well. Yeah. So.
Well, and that Clob, Willardi.
I haven't seen that. That sounds trippy.
Yeah, they were.
It's in the Snakes of Arizona book.
Gotcha.
And they were thinking it was a different species at first.
I might have it.
Yeah. Kind of crazy.
Yeah. I can think of several examples of hybrids that have been, and that's a whole other thing, right? Like, sometimes species hybridize and actually there is a new species that comes of that. Like, think of Darwin's finches, they're the classic example.
Right.
That's another one where do you draw the line between a hybrid and a species that was born of two different species?
Right. Or ones that are adapting in a different niche to prey on a different insect or something, or
right, exactly.
Whatever, different bill types. And when do you call it the different species? Is it just, it's just adapting to its environment, or is it a different, obviously there's genetics that go along with those changes,
right?
But identifying those genetic changes, especially in Darwin's time, was impossible. And did we?
Well, there's a reason I haven't. So, right. Yeah. There's a reason I haven't looked into the whole North American rat snake thing, because I've had enough. Like, it's just continental speciation is just something else.
Right. And there's really good examples in birds too, where they've split bird species that look pretty identical, but I think you were alluding to that, songs and things like that. Similar things where they look the same and they've been identified for years as the same thing, but now we've recognized there are some differences that maybe warrant that split. And obviously some people disagree with that, and some people are for it and against it, and you're going to always have that. But as long as we know what we're talking about and we can use those labels, whether it's like, I saw this here, or, locality type, things, or.
Right. Well, and to me with the birds, it's like, it's we're using some other feature as a proxy for genes. So if two different bird populations make two different songs and don't interbreed, but they diverged 50 years ago, then all right, that's like two different people speaking two different languages, right? We wouldn't argue that there's different species of human. Like that would be preposterous. But then, and this is why I've had to rein myself in a little bit and like view everything within the bubble of reptiles. Because this, again, stemming from the Squamates podcast, I almost went down the cichlid rabbit hole, and like, what? A species can only be 600 years old and still be its own species? What? And I'm just like, nope, got to shut that out. I can't devote that much brain power to that.
Right. And this whole conversation was making me think, I was thinking of it in the context of geckos, but it's other lizards as well, right? Where you have species hybrids that then sexually reproductive species hybrids that then produce precedent genetically. Which is super interesting, right? So they're sufficiently divergent to disrupt meiosis, but they're still viable offspring that then produce asexually.
There's another feature. I have Lepidophyma, and they the population, or the population that the lizards were collected from that are in the hobby are parthenogenic, but not all of them are.
Right.
Yeah, same with the binos, geckos, and the
exactly. Same with the binos. Same with or even something my university is working with are those skinks that some of them are oviparous and some of them are ovoviviparous. Like they're the same species, but
right.
It's
for
just,
now
you can really go down the rabbit hole of different ways to split up things. And
for sure. I guess the bottom line is, as long as you know what I'm talking about and I know what you're talking about, and we can kind of arrive at the same conclusion of the picture of what we're talking about, then I think we're good. And if
especially when we're not in the crazy academic like bickering over, we're just having fun.
Yeah, exactly. And I think that's what takes the pressure off and lets us say, this is garbage, or I like this, or I hate it.
It's a lot easier to be flippant, right?
Exactly. Yeah. Especially when you have very little knowledge. We've got Dunning-Kruger for taxonomy.
We got that for sure.
Yeah. I know. I'm going to my professor and asking him, like, half of the opinions I've come to have been like, he's not even a herp guy. He's a paleontologist, but he's like knows something about evolution. And so I'm like, hey, is this valid? Like I show him, I just email him with different figures from papers I'm reading, because I'm like, I don't understand this. Is this, is this true? And, sometimes it's like, no, that's actually that's not really a great way of doing things. And I'm like, oh, well, now I'm kind of learning how to be critical.
Yeah. Get his opinion on that Antaresia paper.
Yeah.
Unless he was involved in writing it. No, I'm just
no, I don't. He's a mammal guy. So
okay.
Yeah.
Because I mean,
different ball game.
I talked to Zach Loafman about it, and he's like, "Oh yeah, if that was crayfish, they'd be several different species. You know, they wouldn't be sinking them for sure. You know,
yeah. So, I should go back and look at that again, because I'm curious, because the picture in my head I have is like the Children's and most of the Children's and Simpsons formed a clade, and then the ones in the Kimberley formed their own clade, but I don't remember if that's. Or there were 3 clades, and I don't know. I got to go back and look at that.
There were kind of 4 different like groupings, but they said since we can't say that there's no genetic, swapping there, we're just going to sink them all into one. And I'm like, that's the dumbest thing to do with that. I mean, obviously there's 4 different groupings.
And maybe, if they did, if the divergence is such that they form one relatively, maybe if it's a case of, well, if you do this, if we say these are different things, then all of a sudden we have to say, like, I don't know, for example, maybe there's different species of carpet, like, or different species of brettles. I mean, like, maybe if the decision was based off of that premise, maybe that is a case for using subspecies, right? Where it's like, maybe subspecies can be a way to evaluate within species clusters independent of one another, and you don't have to make those concessions of saying, well, if this is a species, then this has to be a species.
Right. Yeah, that's true. I mean, it's the easy way out in some ways, but then at the same time, they went over and named all the different spotted pythons.
Yeah, I know.
New names.
I know.
Like,
that one's a weird one. Which I kind of like. This is where my own bias comes in. I kind of like the idea of there being a Papuan spotted python, but then also, why isn't the Papuan carpet python different? Like, and that's, and theoretically they diverged at the same time. Well,
exactly.
Right. Yeah.
But how different? Species level?
Yeah, exactly. And then you have to be like, okay, how quickly does this evolve compared to this? And it's like,
exactly.
The evolution rate of an anole versus a tortoise is vastly different. And so, and it's, we're trying to answer questions that we just can't know, but well, and I mean, you look at like hominids and apes, oh my God.
I mean, they're so close, but they're obviously different species, and, the genetics are,
and that's, I mean, in the public, in the media, I can think of a recent example being with the whole green anaconda split is like 5% divergence between these 2 anacondas. Humans and chimpanzees are only 2%. Well, this is crazy. And it's, the ill-informed, or I shouldn't say, but like the layperson would think, oh, wow, that is crazy. But, they, when you dig a little deeper and you realize, like, anacondas just probably have been relatively the same for such a long time, and genetic drift has allowed them to have perceived within species diversity, and humans are just a very young species in reference to that.
Yeah. Well, and geckos, I mean, they're really good at finding cryptic diversity in geckos and naming new gecko species. Like, that happens about every week. We were just talking about one of the papers that split up Galeatus into 3 species, and they're all pretty, not too far from each other, but they've got different portholes on the side of them, or
I mean, and when you get into
the genetics are different. So,
it can really do your head in once you really get down to it.
Yeah, I just wish there was more consistency. You had the same.
Right. And that's where in my head I'm like, should we? There's so many. Again, I go back to there's so many different uses for the subspecies rank. Should we just get rid of it altogether? And just accept. But it's something I think about a lot. Yeah. I guess that's the main thing is things are ill structured. There's no good definition of what a species is. I mean, there's lots of different definitions. There's lots of good definitions, but
there's no consensus on what definition we should necessarily use for reptiles or even between different groups of reptiles. And that's that's kind of what's frustrating, I think, to the layperson is we just want the labels we want. We, or at least the labels to agree with, between groups. And that's,
maybe we should just have like accepted common names, like the bird people, like exactly. It's just, this is, yeah.
Yeah. So I don't know. Yeah. Half the time you don't even know what the common name is, you only know the scientific name. I was trying to think like, and I wrote a book on the Nephurus and like, it's like, wait, what's Shai again? What's the?
I was thinking the same thing. I was like, what?
Like Northern Rough, Kimberly Rough. There's a few different ones.
I was messing with the pictures of the one, and I was like, what am I going to post? And then I was like, I looked at yours. I was like, Northern Rough Knobtail. Okay, fair enough.
I think I just defer to A-Rod or whatever that website and just use the same names that they use, or I don't know. I looked on INAT and like they've sunk the Stimson eye into children eye, so they're calling them Stimson's pythons children beyond that. So that's kind of sad to see that go.
And it marks them all out as though you mislabeled it, and that's one of the most irritating things about about INAT records is it's like, this, I don't know, that little feature I don't care for.
Yeah, I mean, I think INAT just goes off of the reptile database, right? And it's they're not. It's a monumental task to document all of current taxonomy and reptiles. So it's
crazy.
I cut them a bit of slack there, but you're absolutely right. Like that definitely can be a little bit frustrating, and especially when you're trying to conserve like scientific literature history, it brings up the argument of why Varanus are all still Varanus and not Euprepiosaurus and Samosaurus and Hapterosaurus and all the different, and a lot of people will argue that's just it's for the sake of literacy in the literature, if that's a thing,
There was an interesting INAT record that I was looking at the other day, kind of going back and forth, and it was an Aspidites, but it had its head chewed off by a cat. And so you just saw the banded pattern. And it's within the range of womas, but it wouldn't necessarily be unexpected that a blackhead might venture just, cause it's like the southernmost tip of the blackhead distribution. So you're like, and it looked like a blackheaded pattern. And then so I'm like, nah, that's a blackhead. And then they said, it's a juvenile. So, that changed, they're a little darker as juveniles. Their bands are darker when they're juveniles. Maybe it is this woma. But the bands sure look blackheaded bands. It was kind of an interesting puzzle sometimes on
and it brings up
with the Aboriginal kid bringing it from up north and coming down and
who knows,
they just ate half of it or something and threw out the other half because it got bad, or I don't who knows.
Exactly. And I think it was Varanus Rainer Guntherei was described for the type specimen was deformed. But they used the deformed snout as a feature to describe it, to say, it's got a bunch of, but it's like, nope, that was just that one individual. Like, I think it's born out of haste for describing things. And that's a bit of a broader scale than say the blackhead, just trying to identify an individual. Yeah. But you're right. It's like a little bit of impatience.
Yeah. Well, maybe. There was somebody, in the morph game or whatever, the heifer bottlenose or whatever they had, really underdeveloped embryo, that they cut open out of the egg and it was white. And they're like, hey, I've got a white snake. I'm like, no, you've got an embryo. You don't have
exactly.
It hasn't developed pigmentation yet,
Yep.
And so we always laugh, it's heifer bottlenose or whatever.
Yeah, right. I know, isn't it like I saw some meme that was like comparing a fetus of a human to a dolphin, and it's like we go through the same steps, we just kind of veer off at the end, know.
Those legs split apart instead of sticking together. The fin. It's fun to think about, and especially from where we're not experts, so it doesn't matter what we play. We can have fun with it and talk about different things, and and I think that's part of the fun of all of this is thinking about what you have in your hands. I was listening to a podcast today, and it was really an interesting podcast, but it was like this guy was talking about species that he's keeping in captivity, and they said, where does that come from? And he's like, oh, I don't know. You're like,
Yeah,
and it was
right.
a rare gecko. Like,
Yeah.
not many people have these. And you're like, you didn't even, you don't even remember where it's from. Like, it's got a very small range and it's very specific for this one area. You're like,
yeah,
come on, do better. Like, let's look at the wild things a little.
Well, that's the
better.
classic, like, yeah, the ball python is from Africa, right?
Yeah.
It's yep, you're right, it is. But how that's the little whatever. I don't know what I'm trying to say. The note card version of it, but yeah. If you just gave somebody that information and they went to Africa and found a python, then all of a sudden an Angolan python's a ball python. It's like,
yeah. I think that's just the get out there and herp kind of thing, get out there.
Oh yeah.
Do stuff in the wild, like experience it how it should be, not just in a deli cup, like.
Yeah. I have to, so I haven't, herping is a very new thing for me.
Mm-hmm.
For the longest time, I wasn't into herping. And then when I didn't get a job this summer, I was like, oh, well, I guess it's time to herp. And so I've been, and then that coincided with me getting into photography a bit. And so now I'm trying to tick off as many salamanders of California as I can.
Yeah.
And it is arguably more rewarding to find that target species than it is to, like, I don't know, get a new animal, for me at least. Maybe I haven't hatched out much stuff, so maybe that can contend with finding that rare target species. But finding my first Dicamptodon giant salamander, like just a trail that I had walked thousands of times in my life, but just had never been there in the right conditions, and all of a sudden there's a pool off the side of the trail, and it's just like a foot long salamander, like. Yeah.
Did you let it bite you?
It was very placid, I have to say. Not nearly as aggressive as some of the arboreal salamanders I've caught.
Yeah. Don't they have pretty good teeth on them?
I've never heard of somebody getting really bit by a dicamp, but the famous photo is of an arboreal salamander with its teeth. And then if you find the photo that's cropped, it's just it's kind of the smiley salamander, and then you find it uncropped and there's a bloody finger above it.
That's awesome. Yeah.
They're super cool.
Yeah. I don't know my salamanders as well as I probably should, but I've seen all the salamanders in Utah. I've found all the salamanders in Utah.
All the forms.
All the one. Yeah. All the one. Tiger salamander. I guess there's kind of two forms, but I don't know.
Yeah.
They're all Marvaritum. So
yeah.
All right. Well, thanks so much for coming on. This has been a really fun discussion, and you brought some really great points out. So really appreciate your viewpoint, and thanks for being here. Where can people find you? Where are you posting your cool photography and salamander finds?
Mostly just Instagram. I'm just all lowercase nick.p.vine. Vine like the plant. And I don't know. I'm just getting into this whole world. So
nice.
I'm not. Yeah.
Yeah, I think I can say the same thing. Like I've just gotten into herping maybe in the last decade or so, seriously. Like, I mean, I was always kind of an outdoorsy kid, always looking for snakes and lizards and stuff, but it wasn't like I was on targeted herping trips. I was just backpacking and, oh, there's a lizard, I'm going to go chase it, or here's a snake, can I bring it home?
There's a concolor.
Yeah, exactly. Here's a baby concolor that I stepped over because it didn't have a rattle, and my dad.
Oh my god! My dad found a Zanata one time, and it was a hike I chose not to go on. I stayed back at the campsite, and then he comes back and shows me a video. He's like, "Hey, I found this cool snake," and it's a Zanata. Yeah. And at the time, I didn't even know what it was, but then looking back, I'm like, okay, a tricolor snake in the Sierras. All right.
Right. Yeah. My dad had a picture from the 60s of him holding a tricolor, and so it was kind of cool. Like, where'd you find it? And then kind of identifying it as a Utah mountain king snake, and it was pretty neat. So, and it was like kind of the northern extent of their range, so he found it pretty high.
Oh wow,
north in Utah, so that was kind of fun too. So I'm just finishing up a field guide to the reptiles and amphibians of Utah, so I'm going to include that picture in the book somewhere.
That's cool.
fit it in just to kind of for the historical perspective. My dad found one before I did, but I wasn't born yet.
Yeah,
so.
right.
Yeah. But yeah, we have some fun stories of herping together and stuff. So
I think it's worse off that Nipper found one before you did.
Right.
Yeah, right.
Coming from overseas. Yeah. Well, see, now this comes back to my roots as a desert rat. Like I wasn't, thinking about mountains as cool places to look for reptiles. So I think I've kind of shot myself in the foot. I still am not sure if I found a green snake in reality or if that was a dream I had, because I have this distinct memory of finding this green snake in a tree, in the mountains somewhere on a trip when I was a kid, and my dad wasn't around. I was just out wandering somewhere, and then I saw it, and then it moved, and then I couldn't see it anymore, and I'm like, oh crap, where'd it go? And I'm all looking through this tree. So did I dream that? Did I see it? I didn't get a picture. So if there's no picture, it didn't happen. Yeah. Speaking of, where do you post your finds?
Photo.
If you don't post them, they didn't happen. Right.
Yeah.
So, yeah. Well, that's cool. Yeah. I'll have to, I don't know if I'm following you on there or not. I'll have to rectify that.
Yeah.
I usually tend only to follow people who post wild reptiles, and if it's captive stuff, I know
I've kind of found myself skewing that way lately too. Yeah.
Right. Yeah. So good times. But I don't know. Usually we end kind of talking about anything cool we've seen in herpetology, any kind of interesting new publications or podcasts you've heard, or interesting new information. I was listening to a podcast. Somebody keeps Naltinus, and they came on a podcast.
Oh yeah,
to talk about it.
I heard that.
Rob recommended that one, so that was pretty cool. And went down, listened to a few episodes on that one, and a few others. But yeah, so that was kind of cool to hear about. And there's some others out there that have them, but since they don't have very many babies very often, like they're just not out there.
Not super public. Yeah.
And if anybody puts it out there, then I'm sure they're just getting mobbed. Like, put me on your list. I want some of those.
Yeah. Right.
Yeah. So
I will never have a list. Yeah. Is basically the answer.
Yeah. Good answer. Yeah. So I don't know. You guys got anything else cool that you've seen recently, or? Yeah.
Not that I can think of. I mean, there's always cool stuff.
Yeah, absolutely. The thing I was going to say last week, but I felt it was too cheesy, was that episode with Jordan. Really was really filling, on my end.
Oh yeah.
He's doing the maps associated with Craig Trumbauer's Pitch book. And so super excited for that. Yeah, absolutely. There was a ton of stuff.
Yeah, I could tell you were digging that conversation after the podcast last week. Jordan's talking, and Rob's just like got this big grin, wide eyed, like, "Oh, this is what I want to hear."
I'm really looking forward to listening to that, because he hasn't done any podcasts, has he?
Yeah, I think I kind of talked him into it, but he was really nice to come on and talk with us about it, and I just wanted to have an excuse to hear his stories from his latest trip, because
oh my god, talk about somebody who's so passionate about about herping, like
right,
sitting at IHS, seeing the photos of the speckled rattlesnakes. That it's just he's seen some crazy stuff. I'm really looking forward to that episode.
Yeah, I've always kind of talked him up, and so Rob was like, "Finally, I get to meet this guy. You keep talking up," and then he's like, "Okay, you delivered. You're the real deal." He wasn't just hyping you. It's real deal. So, but good times. I guess that'll do it. So, thank you for coming on, and thanks everybody for listening. And we'll catch you next week for another episode of Reptile Fight Club.